Before we start, let's have a short recap about our main topic for today, lipids. So what are lipids? Lipids are nonpolar chemical substances that are only soluble in nonpolar solvents and insoluble in water, since water is a polar molecule. These molecules can be produced in the human body and are found in oil, butter, whole milk, cheese, fried meals, and some red meats. Lipids serve a critical role in our bodies. They are the cell membrane structural component. They aid in the production of hormones and provide energy to our bodies. They aid in appropriate meal digestion and absorption. If consumed in sufficient quantities, they constitute a beneficial part of our diet. They are also very significant in signaling.
In this chapter about lipids, we are going to discuss first about the relationship between lipogenesis and citric acid cycle. Lipogenesis is the metabolic mechanism that produces fatty acid from acetyl-CoA. Lipogenesis, like the opposite processes of glycolysis and gluconeogenesis, is not just a reversal of the stages for fatty acid destruction or the beta-oxidation pathway. In general, fatty acid biosynthesis or lipogenesis happens if dietary intake exceeds the amount of resources required for energy production. The liver, adipose tissue, and mammary glands are the principal sites of lipogenesis. During lactation, the mammary glands exhibit enhanced synthetic activity.
The citric acid cycle, on the other hand, is a sequence of biological events in which the acetyl component of acetyl-CoA is oxidized to carbon dioxide, yielding the reduced coenzymes FADH2 and NADH. This cycle, the third step of biological energy production, receives its name from the cycle's first intermediate product, the citric acid. The citric acid cycle is also known as the Krebs cycle, after its discoverer Hans Adolf Krebs, and the tricarboxylic acid cycle, after the three carboxylate groups found in the citric acid cycle.
Except for the succinate dehydrogenase processes that include FAD, the chemical events of the citric acid cycle occur in the mitochondrial matrix, which contains the necessary enzyme. The enzyme responsible for catalyzing this process is a component of the inner mitochondrial membrane. The cycle's component steps are now examined in depth. Four of the 8 processes include oxidation, which yields NADH or FADH2, and 2 involve decarboxylation, which involves shortening a carbon chain by removing a carbon atom as a CO2 molecule. The intermediates in the citric acid cycle's final 4 stages are all C4 molecules: alpha-ketoglutarate, succinyl-CoA, fumarate, and succinate. All of the carbon chains connected to ACP in the first cycle of the 4 repeated reactions in lipogenesis are C4 chains. There are several connections between these two groups of C4 organizations. The last 4 intermediates of the citric acid cycle are related in the following way: saturated C4 diacid, unsaturated C4 diacid, hydroxy C4 diacid, keto C4 diacid. The intermediate C4 carbon chains of lipogenesis are related in the following ways: keto C4 monoacid, hydroxy C4 monoacid, unsaturated C4 monoacid, saturated C4 monoacid. There are 2 significant distinctions in these compound sequences. First, the citric acid intermediates involve C4 diacids and the lipogenesis intermediates involve C4 monoacid. C4 diacids or 4-carbon dicarboxylate acid such as oxaloacetate and alpha-ketoglutarate are involved in the TCA cycle. These intermediates undergo redox reactions that result in the formation of NADH and FADH2. C4 monoacid or 4-carbon monocarboxylic acids such as malate are involved in lipogenesis.
Citrate produced by the TCA cycle is transported out of the cytoplasm and transformed back to oxaloacetate and acetyl-CoA. The acetyl-CoA is subsequently used to synthesize fatty acids, while the oxaloacetate is transformed to malate.
The transition from C4 diacids in the TCA cycle to C4 monoacid in lipogenesis reflects the compartmentalization of these activities as well as the necessity to move intermediates or citrate from mitochondria to cytoplasm. The cytoplasmic conversion of citrate to malate permits the cell to employ the acetyl-CoA generated in the TCA cycle for fatty acid synthesis, thereby satisfying the demands of lipid production in a separate cellular compartment. Second, in lipogenesis, the various acid derivative types are encountered in the reverse sequence that they are seen in the citric acid cycle, while the citric acid cycle proceeds in the order citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate. Lipogenesis frequently proceeds in the reverse order, with an emphasis on fatty acid synthesis from acetyl-CoA and malonyl-CoA.
Fate of fatty acid-generated acetyl-CoA. The fate of acetyl-CoA is very dynamic and is determined by the cell's metabolic state, food availability, and energy requirements. Cells can adapt to changing environments and maintain energy homeostasis by integrating these pathways. So what happens to the acetyl-CoA formed as a result of fatty acid oxidation? It can choose from a number of various possibilities. Among these alternatives are the following. First is the acetyl-CoA can be further processed through the common metabolic pathway to obtain ATP. 2. The acetyl-CoA can undergo conversion to ketone bodies. Such ketone bodies can be reconverted when needed back to acetyl-CoA, which can then be processed for ATP production. The acetyl-CoA can be stored in the body in the form of triacylglycerols. By reconverting via lipogenesis the acetyl-CoA to the fatty acids that are then converted to triacylglycerols. The acetyl-CoA can be used as a starting material for the production of lipids, other than fatty acids that the body needs. A specific example of such other lipids is production of biosynthesis of cholesterol. As was previously discussed, cholesterol is a necessary component of cell membranes. It is also the precursor for bile salts, sex hormones, and adrenal hormones. The biosynthesis of cholesterol, a C27 molecule, occurs primarily in the liver. Biosynthesis of cholesterol consumes 18 molecules of acetyl CoA and involves at least 27 separate enzymatic steps. The rate-determining step in the formation of cholesterol occurs early in its biosynthesis. It involves the formation of the C6 intermediate mevalonate. In a multi-step sequence, 3 acetyl-CoA molecules are condensed together to produce the C6 species. Extensive research has been carried out concerning this slow step, with the goal being prevention of its occurrence as a means of decreasing the body's internal production of cholesterol in order to decrease blood cholesterol levels. It is also important to note what cannot happen to the acetyl-CoA obtained from fatty acid oxidation. In humans and animals, it cannot be used for the net synthesis of glucose. Pyruvate or oxaloacetate is the needed starting material for the glucose production via gluconeogenesis. Human and animals do not possess the enzymes needed to convert acetyl-CoA to pyruvate. By contrast, plants do obtain the 2 additional enzymes needed. Pyruvate can be converted to acetyl-CoA in a reaction sequence that is irreversible. Acetyl-CoA cannot be converted to pyruvate in step 1 of gluconeogenesis. Pyruvate is converted to oxaloacetate, and oxaloacetate is also produced in the last reaction of the citric acid cycle. However, use of such oxaloacetate in glucose production cannot lead to a net increase in glucose. In each round of the citric acid cycle, 2 carbon atoms enter the cycle as acetyl-CoA and 2 carbon atoms leave the cycle as CO2. Thus, there is no gain in carbon atoms in turn of the cycle. The oxaloacetate produced in the last step of the cycle is not newly formed oxaloacetate, but rather regenerated oxaloacetate in step 1 of the citric acid cycle. Oxaloacetate is consumed. And in step 8, it is regenerated, thus no net increase on oxaloacetate production occurs during the citric acid cycle because there is no net increase in carbon atoms processed. As a result, no net increase in glucose production can occur using oxaloacetate.
Relationships between lipids and carbohydrate metabolism. The primary link between lipid and carbohydrate metabolism is acetyl-CoA. Interrelationships between carbohydrate and lipid metabolism. Acetyl-CoA is a byproduct of the breakdown of glucose, glycerol, and fatty acids, as well as a starting material for the production of fatty acids, cholesterol, and ketone bodies. Lipids and carbohydrates are two major types of macromolecules that play critical functions in living organism metabolism. The interactions between lipids and carbohydrate metabolism are complicated and linked, as both are required for energy balance and different psychological processes. The following are some of the most important links and interaction between lipid and carbohydrate metabolism. Carbohydrates, such as glucose, are the body's principal short-term energy source in terms of energy storage and utilization. Glucose is easily accessible and can be rapidly digested to produce ATP via glycolysis. When digested, lipids serve as a mere efficient long-term energy storage molecule because they provide a higher energy yield per gram. Excess energy, particularly glucose, can be converted to lipids and stored as triglycerides in adipose tissue. In terms of glucose and fatty acid interconversion, excess glucose in the liver can be turned into fatty acid by a process called de novo lipogenesis. This is a method for the body to store extra energy from carbs as lipids, whereas gluconeogenesis can convert fatty acids to glucose in specific circumstances, such as starvation or low carbohydrate consumption. This mechanism is mostly carried out in the liver to keep blood glucose levels stable. Insulin, a hormone secreted in responses to rising blood glucose level, increases glucose uptake by cells for energy synthesis and storage. It also suppresses gluconeogenesis. Insulin has an effect on lipid metabolism as well. It enhances fatty acid storage, triglycerides in adipose tissue, and inhibits lipolysis, which is the breakdown of triglycerides into fatty acids. In terms of ketonegenesis and ketone bodies, the body switches to alternative energy sources during periods of low carbohydrate intake or fasting. In the liver, fatty acids are oxidized to form ketone bodies, a process known as ketogenesis. While using ketone bodies such as oxytoacetate, beta-hydroxybutyrate can be used as an energy source by numerous tissues, including the brain when glucose availability is low. Certain lipid molecules such as phospholipid and sphingolipids act as signaling molecules, regulating numerous cellular processes, including those related metabolism. Insulin signaling. Lipids participate in insulin signaling pathways, altering cell responses to insulin and hence influencing glucose and lipid metabolism. To summarize, lipid and carbohydrate metabolism is tightly linked, with both playing critical roles in energy storage consumption and overall metabolic balance. These processes are dynamically regulated by the body in order to respond to shifting energy demands and nutrient availability.
B vitamins and lipids metabolism. 4 of the 8 B vitamins are required for the processes of beta oxidation, ketogenesis, and lipogenesis. Niacin, riboflavin, pantothenic acid, and biotin are examples. First and foremost, the B group vitamins are a collection of 8 water-soluble vitamins that are required for a variety of metabolic processes. B vitamins, which include B1, B2, B3, B5, B6, B7, B9, and B12, work together to help with energy metabolism, DNA synthesis, red blood cell creation, and neurological function. These vitamins help in energy metabolism, DNA synthesis, red blood cell creation, and neuron function. Their function is to maintain overall health, promote healthy growth, and prevent deficits that could lead to disease. Now, let us discuss the 4 of the 8 B vitamins that are required for the processes of beta oxidation, ketogenesis, and lipogenesis. Niacin, often known as vitamin B3, as a water-soluble B vitamin that is naturally present in certain foods, added to foods, and supplied as a supplement. Nicotinic acid and nicotinamide are the two most frequent types of niacin found in food and supplements. Niacin is a precursor of the coenzymes nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate, both of which are important in redox processes. NAD participates in both lipolysis and fatty acid production. Number 2. Riboflavin. Vitamin B2, often known as riboflavin, is one of the 8 B vitamins. All B vitamins aid the body in the conversion of food, which is then utilized to generate energy. These B vitamins, often known as B-complex vitamins, assist the body digest fats and proteins. B vitamins are required for a healthy liver, skin, hair, and eyes. They also aid in the healthy functioning of the nervous system. Riboflavin is a coenzyme precursor for the redox coenzymes flavin mononucleotide and flavin adenine dinucleotide. FAD is a cofactor for enzymes involved in fatty acid beta oxidation, which occurs when fatty acids are broken down in the mitochondria to create acetyl-CoA. Pantothenic acid. Pantothenic acid is an essential nutrient found naturally in some foods, added to others, and accessible as a dietary supplement. This water-soluble B vitamin is primarily involved in the formation of coenzyme and acyl carrier protein. Pantothenic acid is a component of coenzyme A, which is essential for fatty acid synthesis and degradation. CoA is required for fatty acid activation before they enter the mitochondria for beta oxidation. Number 4. Biotin. You might be familiar with vitamin B7 as biotin. It is a water-soluble B vitamin that can be found in various foods as well as supplements. Biotin is essential for enzymes to break down lipids, carbs, and proteins in diet. It also aids in the regulation of cell signaling and gene activity. Biotin is a cofactor for carboxylase enzymes such as acetyl-CoA carboxylase, which converts acetyl-CoA to malonyl-CoA, and so plays an important part in fatty acid production. Now, what is lipid metabolism and how does B vitamins interrelate to it? Lipid metabolism is the process by which lipids are synthesized and degraded in cells, involving the breakdown or storage of fats for energy. The oxidation of fatty acids to create energy or to manufacture new lipids from smaller constituent molecules is what lipid metabolism is all about. Lipid metabolism is involved in many important bodily functions, including energy storage, hormone regulation, nerve impulse, transmission, and fat-soluble nutrient delivery. Lipids are a source of energy. In conclusion, B vitamins play an important role in lipid metabolism by acting as a cofactor for enzymes involved in critical metabolic events. They aid in the synthesis, degradation, and consumption of lipids, as well as the maintenance of cellular energy balance and the support of different physiological activities.